Overview of the Krebs cycle. The German-British scientist Hans Krebs discovered the Krebs cycle in the 1930s. Thus, in honor of his discovery and pioneering study, the pathway was named after him. The Krebs cycle is also known as the citric acid cycle or tricarboxylic acid cycle because citric acid is its first product, which is composed of three carboxyl groups, or the COO. In eukaryotic organism, the Krebs cycle occurs in the mitochondrion. Eukaryotic organisms are organisms whose cells have a nucleus enclosed within a nuclear envelope, while the mitochondrion is a double membrane-bound organelle found in the most eukaryotic organisms. The citric acid is composed of three carboxyl groups, or the COO. It is the first product of the Krebs cycle, which forms from the reaction between acetyl-CoA and oxaloacetate. The previous lesson dealt with the mechanism of glycolysis. This chapter emphasized that the final products of glycolysis are two pyruvate molecules. These molecules carry a lot of energy in them that they still need to undergo a series of processes to maximize energy generation. In the presence of oxygen, the pyruvate produced from glycolysis undergoes the Krebs cycle. The carboxyl group consists of a carbon bonded to both an oxygen and a hydroxyl group, while the acetyl-CoA or acetyl coenzyme A is a molecule that participates in many biochemical reactions in protein, carbohydrate, and lipid metabolism. Its main function is to deliver the acetyl group to the citric acid cycle or the Krebs cycle to be oxidized for energy production. Pyruvate enters the mitochondrion, where it has to be oxidized first before entering the Krebs cycle. The resulting molecule from the oxidation of pyruvate, acetyl-CoA, enters the Krebs cycle and undergoes a series of oxidation and reduction. As a result, these processes yield carbon dioxide and the electron carriers NADH and FADH2. So first, what is pyruvate? Pyruvate is an important chemical compound in biochemistry. It is the output of the metabolism of glucose known as glycolysis. One molecule of glucose breaks down into molecules of pyruvate, which are then used to provide further energy in one of two ways. While what is the NADH and FADH2? Both of them are high energy electron carrier used to transport electrons generated in glycolysis and Krebs cycle into the electron transport chain. And what do they do? These two compounds are created after the Krebs cycle in respiration and are vital in helping to transport electrons into the mitochondria, which are used them in the electron transport chain. They are energy molecules that are utilized by the cell to continue the process of cellular respiration. The Krebs cycle starts when acetyl-CoA reacts with oxaloacetic acid, also known as oxaloacetate. As a result, citric acid or citrate is formed. Citric acid undergoes a series of oxidation and reduction which results in the formation of CO2, NADH, and FADH2. Like what I've said earlier, the Krebs cycle, also known as the citric acid cycle or the tricarboxylic acid cycle, is one of the most important reaction sequences of biochemistry. And also, the acetyl-CoA reacts in the first step of the 8-step sequence of reactions that comprise the Krebs cycle, all of which occur inside the mitochondria of eukaryotic cells. While the Krebs cycle does not produce carbon dioxide, this cycle does not produce significant chemical energy in the form of ATP directly. And also, this reaction sequence does not require any oxygen. Instead, this cycle produces NADH and FADH2, which feed into the respiratory cycle, also located inside of the mitochondria. It is the respiratory cycle that is responsible for production of large quantities of ATP and consumption of oxygen. How is acetyl-CoA or acetyl coenzyme A formed from pyruvate? transition reaction, pyruvate, a 3-carbon molecule, cannot immediately enter the Krebs cycle. It must first undergo oxidation to become another molecule that can enter the pathway. After glycolysis, pyruvate molecules enter the mitochondrion, given that the condition is aerobic. Upon entering, pyruvate is converted into acetyl-CoA. This process starts when the carboxyl group or the COO in pyruvate is removed and released as carbon dioxide or CO2. How is pyruvate converted to acetyl-CoA? Pyruvate is converted into acetyl-CoA in an intermediate process just before the citric acid cycle. Here it reacts with coenzyme A. Here it loses 2 of its oxygen and 1 of its carbon to form carbon dioxide, and then the resulting molecule is a molecule of acetyl-CoA. Carboxylation also results in the formation of a 2-carbon molecule. Then this 2-carbon molecule undergoes oxidation and becomes acetate. The electrons released from the oxidation of a 2-carbon molecule are transferred to NAD+, which then becomes NADH. The acetate molecules react with a sulfur-containing compound called coenzyme A. As a result, acetyl-CoA is formed. The whole process of pyruvate oxidation or transition reaction is catalyzed by an enzyme called pyruvate dehydrogenase. And as you know, acetyl-CoA enters in the TCA cycle where it is oxidized for energy production. The transition reaction between glycolysis and Krebs cycle converts pyruvate to acetyl-CoA through oxidation. This process releases a carboxyl group along with a reaction with coenzyme A. Acetyl-CoA is generated either by oxidative decarboxylation of pyruvate from glycolysis, tulad nga po sa definition. And then acetyl-CoA then enters in the TCA cycle where it is oxidized for energy production. At kung makikita niyo po rito sa figure, nagmumula siya kay pyruvate na kung saan kinuwanan si pyruvate ng carbon dioxide or the CO2. And then after that, dito sa may bandang gitna, hiniwalay po si carbon kay oxygen. And then the coenzyme A is added, which is the S is equals to COA. And then after po nun, NAD plus will become into NADH. Just like what I've said po kanina or sa in-explain ko po sa mga past slides, NADH po ay isang high energy electron carrier na ginagamit sa pagtatransport ng electrons generated na sa glycolysis at Krebs cycles papunta po sa electron transport chain. At kapag nangyari na si NADH, ay dagdagan na rin siya ni coenzyme A, ayun na po ang last stage para makakuha tayo ng acetyl-CoA. Mechanism of the Krebs cycle. Krebs cycle starts when the acetyl-CoA from the transition reaction, a 2-carbon molecule, reacts with oxaloacetate, a 4-carbon molecule, resulting in the formation of citric acid or citrate, a 6-carbon molecule. This process is catalyzed by citrate synthase. Citrate undergoes isomerization and becomes isocitrate via enzyme aconitase. Isomerization involves the rearrangement of the atoms of citrate, which results in the formation of its isomery citrate. Then, isocitrate undergoes oxidation, which results in the formation of alpha ketoglutarate, a 5-carbon molecule. The oxidation of isocitrate causes the release of carbon dioxide and the reduction of NAD plus to NADH. This process is catalyzed by isocitrate dehydrogenase. Citrate is formed from the reaction of acetyl-CoA and oxaloacetate. Citrate undergoes isomerization and becomes isocitrate. Then isocitrate is oxidized to produce an alpha-ketoglutarate. As you can see at the figure out here, the left corner, it has 8 processes all in all. First one is citrate, second isocitrate, third alpha ketoglutarate, fourth succinyl-CoA, fifth succinate, sixth fumarate, seven malate, and eight, or the last one, is the oxaloacetate. And then as the result of the Krebs cycle, all in all, you will get 3 NADH, 1 FADH2, 1 GTP, 2 CO2, or the carbon dioxide. But Krebs cycle repeated itself twice. So what you will get all in all is 6 NADH, 2 FADH2, 2 GTP, and 4 carbon dioxide. And then the electron transport chain will get you the ATP, or what we call the adenosine triphosphate.
So next, where does CO2 go after being released from the Krebs cycle? The next step involves the oxidation of A-ketoglutarate, which is catalyzed by A-ketoglutarate dehydrogenase. Carbon dioxide is released and NAD reduced to NADH during this process, which leaves a 4-carbon molecule behind. So, ang oxidation, ito ay isang biological process that involves the loss of electrons. So, ang sinasabi dito, ang A-ketoglutarate goes the process of losing electrons, which generates an A-ketoglutarate dehydrogenase. So, pagkatapos, ang carbon dioxide dito ay ini-release siya, at sa proseso na ito, nag-reduce ang NAD to NADH. Kaya naiwanan ng 4 carbon molecules. The 4 carbon molecule then reacts with coenzyme A, which forms the succinyl-CoA. This process is catalyzed by succinyl-CoA synthesis. Afterward, the coenzyme A formed succinyl-CoA is replaced by a phosphate group. However, the phosphate group is immediately transferred to GTP, which is guanosine diphosphate, to form GTP, which is guanosine triphosphate, a molecule similar to ATP in terms of function. This process results in the formation of the 4 carbon molecule succinate. So, pagkatapos, nag-respond ng 4 carbon molecule sa coenzyme A, which is an acetate molecule that reacts with sulfur-containing compound, forms a succinyl-CoA, then generates succinyl-CoA synthesis. So, pagkatapos nun ay ang succinyl-CoA is changed to phosphate group, which is transferred to GTP para magka-form ng GTP. Kaya, yung 4 na 4 carbon molecule succinate ang naging result sa process na ito. When A-ketoglutarate is oxidized, it releases carbon dioxide and causes NAD+ to reduce to NADH. Succinyl-CoA is the result of the oxidation of A-ketoglutarate and reaction with coenzyme A. Then the coenzyme A in succinyl-CoA is replaced by a phosphate group, which results in the formation of succinate. So, sa slide nito, ito lang yung parang kabubuan ng last na slides na na-discuss. So, kapag ang A-ketoglutarate ay na-oxidize, nagre-release ng carbon dioxide or CO2, at ang NAD+ ay nagle-lessen to NADH. Tapos, ang succinyl-CoA naman ay ang resulta ng oxidation of A-ketoglutarate at reaction ng coenzyme A. So, pagkatapos, then coenzyme A is replaced by phosphate group that forms the 4 carbon molecule succinate. Succinate, a 4 carbon molecule, is oxidized to form fumarate, another 4 carbon molecule. The oxidation of succinate to produce fumarate causes 2 hydrogen molecules to be released from succinate. These hydrogen molecules are transferred to fat, which is reduced to form FADH2. This process is catalyzed by succinate dehydrogenase. Afterward, the fumarate receives a water molecule, which results in the formation of malate, another 4 carbon molecule. The addition of water to fumarate is catalyzed by fumarase. Lastly, malate is oxidized and becomes oxaloacetate. The oxidation of malate causes the reduction of NAD+ to NADH. This process is catalyzed malate dehydrogenase. When oxaloacetate reacts with acetyl-CoA, another round of Krebs cycle takes place. Thus, one cycle always regenerates oxaloacetate. So, yung succinate is oxidized to form fumarate. So, yung fumarate, ito naman yung another 4-carbon molecule. So, since inoxidized ang succinate to fumarate, which causes 2 hydrogen molecules to be released, that those 2 hydrogen molecules are transferred to fat, which is flavin adenine dinucleotide, that lessens it to fat H2. Pagkatapos, ang fumarate ay makaka-receive ng water molecule which forms malate, then yung addition water is catalyzed to fumarate to fumarase. When malate is oxidized, magiging oxaloacetate siya. So, this oxidation also causes the reduction of NAD+ to NADH. This process is catalyzed malate dehydrogenase. So, pag nag-reacts ang oxaloacetate sa acetyl-CoA, uulit na naman ang Kreb cycle. So, sa isang cycle, they always generate or to regrow oxaloacetate. Susunod, succinate undergoes oxidation to form fumarate. The addition of a water molecule to fumarate forms malate, then malate oxidation forms oxaloacetate. So, parang kabuuan lang to ng last slide. So, pagka ang succinate ay inoxidize, mag-form siya ng fumarate, then add water molecule to form malate, then oxidize it to form oxaloacetate. Much energy is produced in the Kreb cycle. The process of the Kreb cycle starts after the conversion of pyruvate into acetyl-CoA. It should be noted that after glycolysis, every glucose molecule yields 2 pyruvate molecules. Thus, the Kreb cycle happens twice for each round of glycolysis. In this case, all products of the Kreb cycle discussed are doubled to account for every glucose molecule that undergoes glycolysis. The product of the transition reaction and the Kreb cycle are summarized in Table 9.3.1. So, sinasabi dito, ang Kreb cycle ay nagsimula pagkatapos ng conversion ng pyruvate sa acetyl-CoA. And after glycolysis, dalawang glucose molecule ay mag-yield ng dalawang pyruvate molecules. So, sa round ng glycolysis, nangyayari itong ng dalawang beses sa Kreb cycle. Tapos, all products of the Kreb cycle are doubled to account for every glucose molecule. So, dito, ang summary of the products of the transition reaction and the Kreb cycle. So, sa lahat ng process involved ay dalawa lang na molecules ang napoproduce nila. So, pagdating sa oxidation of pyruvate to acetyl-CoA, oxidation of isocitrate to A-ketoglutarate at oxidation of A-ketoglutarate to succinyl-CoA ay CO2 or carbon dioxide at NADH, nicotinamide adenine dinucleotide hydrogen ang products nila. So, pagdating naman sa oxidation of succinate to fumarate, ang products niya ay FADH2, flavin adenine dinucleotide. Tapos, ang release of a phosphate group from succinyl-CoA IGTP guanosine triphosphate. Tapos, ang last ay ang oxidation of malate to oxaloacetate ay NADH din. As per table 9.3.1, the transition reaction yields 2 molecules of CO2 and 2 molecules of NADH. In the Kreb cycle, there are a total of 6 NADH molecules, 4 carbon dioxide, and 2 FADH molecules produced. These molecules are produced from the oxidation of pyruvate, isocitrate, A-ketoglutarate, succinate, and malate. The NADH and NADH2 FADH2 molecules will enter the next stage of cellular respiration, the electron transport chain, while CO2 is released to the environment to respiratory organs such as lungs. The GTP is produced in the Kreb cycle has the same function as the ATP. Thus, it can be considered equivalent to 2 ATP molecules produced and are ready to be used by the cells. Table 9.3.2 summarizes the reactant and products of the transition reaction and the Krebs cycle. So sa Krebs cycle, a total of 6 NADH molecules at 4 na carbon dioxide and 2 FADH2 molecules ang naproproduce. So they were produced by the oxidation of the pyruvate, isocitrate, and acetoglutarate, at succinate at saka malate. Tapos, the NADH and FADH2 molecules ay papasok sa susunod na stage of cellular respiration, which is the electron transport chain. CO2 is then released to the environment through organs like the lungs. And also, GTP produced has the same function as the ATP. Reactants and products in the transition reaction in the Krebs cycle for every glucose molecule. So, dito makikita natin ang process, which is the transition reaction at saka Krebs cycle. Tapos, ito yung overall reaction niya. So, sa transition reaction, ang overall reaction niya is 2 pyruvate plus 2 coenzyme A plus 2 NAD plus becomes 2 acetyl-CoA plus 2 CO2, which is carbon dioxide, plus 2 NADH. So, sa Krebs cycle naman, ang overall reaction niya is 2 acetyl-CoA plus 6 NAD plus 2 fat plus 2 ADP plus 2 P becomes 4 CO2 plus 2 CoA plus 6 NADH plus 2 FADH plus 2 GTP. Dito sa slide na to, makikita natin mga facts lang. So, did you know the formation of GTP or ATP as the result of the release of a phosphate group from succinyl-CoA depends on the type of cell. In humans, GTP is the form that is mainly produced in liver cells, while ATP is the one produced in heart cells. However, it should be noted that this one is only true in the Krebs cycle and not in the other stages of cellular respiration.
And that's all for today. Thank you for listening.